Good evening and welcome to Genomics Lite. This is our careers session, talking to three different researchers from the Wellcome Genome Campus about their careers and work in cancer research. My name's Em, I'm the Education and Learning Officer at the Wellcome Genome Campus, so I'll be hosting the session and helping out with any help anyone might need. We've also got Fran on the call who will be helping out as well. Before we get started, I'm going to let our three speakers very quickly introduce themselves, and then I'll explain how the session's going to work. So Patricia, do you want to have a quickly introduce yourself?
Sure. Hi, I'm Patricia Yax. I'm a staff scientist at the Institute, and you'll learn a little bit more about my career path and how it is to become a computational biologist if you're lab worker before.
Awesome. Hayley, would you like to introduce yourself?
Hello, everyone. I'm Hayley Francis, and I'm a principal staff scientist at the Wellcome Sanger Institute.
Wonderful. And Jose?
My name is Jose. I am a PhD student at EMBL EVI, which is the European Bioinformatics Institute, doing cancer data science.
Awesome. So we've got three quite different speakers with us this evening who've done different aspects of cancer research. So to start with, for those of you who might not have joined us this evening, we host all of these events in the spirit of curiosity. So we ask that you join us in that spirit of curiosity this evening, respecting everyone's different views, identities and experiences. Myself and Fran are here to moderate the platform to make sure it's a safe and inclusive space for everyone joining us. In case you've not used Zoom webinar, you'll have spotted you can't use your camera or your microphone this evening. So if you need to get in touch with us, there's a few different ways you can do that. You can use the chat box just to generally say hello or to answer any questions that the panelists might generally ask. If you have specific questions for the panelists about their career, about anything they talk about in their talk, or more broadly about what it's like to work in biomedicine and in cancer research, you can use the Q&A button at the bottom to submit your questions, and we'll pull some out in between each speaker and more at the end as well. We've also got closed captions available if you'd like them. If you press the little CC box, they should jump up on your screen. You can also move them around the screen. So if you need to move them so you can read text on someone's slides, just move them out of the way. Likewise, you can hide them if you don't want them. This session is being recorded, so if you can't save for the entire thing, it'll be available on our website and on our YouTube channel, probably tomorrow, if not early next week. This is the last in our cancer research series, but the previous ones are all available again on our website and YouTube channel as well to catch up. So all five of us in normal times would be on the Wellcome Genome Campus, and actually Hayley is on the campus at the moment, which is a rarity for these talks. The Wellcome Genome Campus is just south of Cambridge in the UK, so it'd be great to know whereabouts in the world everyone's joining us from this evening. We've got one person from London, so I always like to see who's furthest away from campus. So London's winning by default so far, but let us know in the chat box where you're logging in from. Kettering, I think London still might be further, so we'll see who else we've got. Oh, from the Wirral, so we've got quite a UK base this today. Turkey. Okay, I think Turkey's winning so far. On the campus then, there's two main research institutes. There's the Wellcome Sanger Institute and there's EMBL EBI. Everything that everyone does on campus has something to do with that genome word in our title. So we all research something to do with DNA. Wellcome Sanger combines wet lab, so using pipettes and chemicals with data analysis, and EMBL EBI focuses on just data analysis. And you'll hear a little bit more about both halves of that story from our speakers tonight. We cover lots of different topics on campus, from human genetics, so looking at how diseases are passed down families, obviously cancer, which is the topic of this series, how cells work, parasites and microbes with a big focus on COVID at the moment, the tree of life, looking at how life is linked together, and biodata, which we'll also touch on in the talk today. Before I hand back over to our speakers, if you are studying for your GCSEs, A-levels, or any sort of high school or university level qualifications, our Your Genome website is a really great resource full of videos, articles, activities all around genomics. And you can sign up to our mailing list to find out when we've got new events and new opportunities happening. And just to highlight a couple of events that are sort of coming up, our team is organising these COVID connection talks all about genomics and the pandemic. There's a series of 4 of them. The first one was last week, and they'll continue throughout the rest of the year. And for anyone interested in sort of biodiversity and ecology, our Darwin Tree of Life team are currently hosting activities and live chats on their platform. All of these links I'll pop in the chat box in a minute and in the emails you'll get from Eventbrite tomorrow, so you won't miss out on any of this. That is all from me, so I will stop sharing my screen, and I think Patricia is our first speaker, so I'll hand over to her. Remember to put any questions for Patricia in the chat box or Q&A as we go.
Hello everyone, I'm Patricia. I work at the Sanger Institute and I work in cancer research. I look at drug combinations and which drug combination might work best for a certain cancer type. Today, I'm going to tell you a little bit about where I come from. I'm not British, I'm German, and how I got here and where I might be moving on soon. And just to give you a feeling for what it might take to get into such a job. So this is my overview of where I've been before. I'm German, as I said, I'm from Northern Germany. And when it came to choosing where to do my bachelor's, it was fairly easy for me because I didn't have the money to move out of my home. So I stayed at home, which is in Hamburg, very good university. So I was quite lucky. So I could stay at home. But then someone told me about, oh, you know, I've been for my master's in London. I was like, oh my God, this would be awesome. So I looked into it and I was very, very happy that I got a scholarship. So I went to London for my master's. And after that, I was like, okay, I'd like to do a PhD. I don't really know where. Let's look into some options. And I decided to go to Switzerland. And to be honest, what made me decide to go to Switzerland was when I was invited for interviews. I walked through the city and I came past the lake that is in the middle of the city center, and it was turquoise and the sun was shining. I was like, this is amazing. This is where I want to be. So I spent 5 years in Switzerland. And then after that, it was really, I knew that coming back to the UK would be a good choice in terms of research, because particularly around London, Cambridge, Oxford, you have so much research happening. And if you want to become someone who leads their own group or moves up the career ladder in science, then having spent a good amount of time in an English-speaking country like the UK or the US is almost a must in almost all other countries. So I knew I had to come back. And I was very lucky that I got the job that I have now. I started as a postdoc, but then took on a lot more responsibilities and moved into a staff scientist position. And now coming to 2021, quite a few years into my journey, I have decided that I would like to move on into industry. And because my husband has been offered a job in Basel in Switzerland, we're probably going to go back. So this is, for me, it's a big question mark. I don't know where I'm going to end up. I just know the country. So let's see how I'm going to end up there. But as you can see underneath the timeline, it's really different topics that have come through the different jobs, which is really nice. So it started very broad. I studied molecular life sciences, which is biology and chemistry. Then I moved into more medical topics and then cancer biology for my PhD. What I do now is really focused on big data. So learning how to explore big data. And through that, like everything that I've done in cancer research, I've become more interested in how it translates into clinics. That's why I want to move into industry, to be closer to the patient, to make a difference. Now, I thought what might be interesting as well are a couple of figures for you. I was very lucky because I had a scholarship for my master's and I did my bachelor's in Germany. I had almost no tuition fees to pay. And that is something to consider for you as well. If you speak English, you can study almost anywhere in the world. And some countries have almost no tuition fees. So that is, I think, a good one to consider. The other thing that you might not be aware of is obviously you get a PhD salary in the UK as well, but it's often quite modest. Whereas in Switzerland, you're paid as if you had a good job. And that translates to roughly £30,000 a year. And you can live very well from that. So even for a PhD, there are different options than the UK. And if you go into science, English is the language that you go for anyways. Everyone speaks English. So that should not be a barrier. So please have a look at different options. And obviously, if you love to stay in the UK, it's a great place to be, but there are other options. Now, I mentioned in my introduction that by training, I'm a lab scientist. So I do a lot of lab work, like treating cell lines with drugs, which we then incubate for a couple of days and we check how many of them are still alive. But through the project that I'm doing now, I've learned a lot of data analysis and on big data. So I've learned a good skill set of computational biology. And that is something, if you like working on a computer, if you like looking into numbers and like playing a numbers detective, then computational biology could be the right thing for you. And if you enjoy it, you will always have a job in research and even outside of research. So please go for it. It's a lot of fun. And a couple of things why I love my job. One is I'm a very curious person, so I like to ask questions and now I can answer them, which is great. In science, depends on the project a little bit how the proportions are, but you usually work on your own, but also in a team. So there's always someone to go to, and it never feels as if you're on your own, which is actually quite nice. Every day there are challenges, and you have to find creative solutions. So if you're a creative mind, that could be a good thing for you. It is fairly flexible working hours. So it doesn't mean that you don't work a lot, and sometimes you work more than you should. But also it means that if you have to pick up your children at a certain time on a day, you usually can make it. So that is not really an issue. So that is actually quite nice. And I love to travel. And through my work, I can go to conferences if it's not COVID times, I have to say. So conferences happen from home these days, but before we could travel all around the world, perhaps once or twice a year, if not more often. So that is actually quite nice as well. That is it for me. I'm happy to take any questions that you might have. Just let me know.
Awesome. Thank you, Patricia. So if anyone's got any questions specifically about Patricia's sort of career progression, pop it in the Q&A or the chat box. Whilst we just have a look if any come in, you've obviously sort of grew up in Germany, worked here in the UK and in Switzerland. If you got to pick any country in the world to go and work in, is there one that you'd really like to work in?
I think I would have to take not only working conditions and like surrounding for work into account, but like quality of living. And that's why we decided to go back to Switzerland, because quality of living is very high in Switzerland. Just being out in the nature, going hiking on the weekend, which is just fantastic. The UK is a little bit too flat for my taste. So I'm quite looking forward to go back. And it's just, to be honest, personal things as well, because Switzerland is going to be back on the continent. So going back home to Germany, we can just jump into a car instead of having to fly home, which is very difficult under a pandemic. Yeah.
Awesome. There's been a couple of questions. One asking, what type of research do you conduct? So what's the sort of nature of your research?
So we look at drug combinations. We have collected data for over 2,000 drug combinations in breast, colon, and pancreas cancer. And we really want to know which of the drug combinations works best, for example, in breast or in colon cancer. So that is what I've been studying in the past couple of years. And we have found quite some exciting stuff. So it all starts with generating the data, which is a big lab team who does that. Then I look into all the numbers, trying to figure out what works well. And then I go back into the lab and do some more experiments to verify that that is a true effect.
Awesome. And then one last one for you, and then we'll move to our next speaker, but there'll be time for more questions for Patricia at the end. Someone's asked whether you enjoyed your time at Imperial when you did your master's. I
did. I quite enjoyed it. It was slightly different than what I was used to in Germany. I lived on my own for the first time, for example. I love London. We always came back every year on holidays because we love it so much. And now we're around the corner. Imperial College it was a slightly different structure than what I was used to as well. So it was less practical than what it is being taught in Germany. Obviously, I had my master's project, which was, I think, 5 months in the lab. I quite enjoyed that, but the whole lecture structure is slightly different than what I was used to. But I met very, very good lecturers. So I guess it depends on which degree you're pursuing, but I really like mine.
Awesome. And I think a big thing that's come from everything you've said is about enjoying the place where you're working as well as the actual institution.
Definitely. And perhaps one thing to add, because that is commented on from students who come from the UK, that often they're lacking practical experience. So if you think that you might like a certain career and you have a couple of weeks on your hands in summer, just try to find an internship and go for it, because that is really make you stand out from the rest of the people. And at the same time, you can see whether you really like it or not. Because I've done quite a lot of internships and I've encountered some places that I didn't like. And that was just as valuable as the ones that I liked.
Awesome. Thanks very much. We'll move on to our next speaker for now. So Hayley, if you want to share your slides, and we'll hear from Hayley next.
There we go. Hopefully you guys can see my slides.
Yeah, that's looking great.
Perfect. Sorry, 1 second. Okay, so hello to you all. This is the first time that I've participated in one of these career panels, so hopefully I have something interesting to say to you guys. So I guess just to sort of kick off, I sort of started life in Wales. So I was born and raised in Wales, and completely loved my time there. This is going back and forth. Why are you doing this to me?
Yeah, that looks great.
There we go. Yeah, so I was born and raised in Wales. I'm from a very small Welsh village in sort of the coal mining valleys, and I was the first person in my family to go to university. And I think sort of out of that, you sort of get a little bit confused about what you want to do. And I always sort of loved biology. I had sort of like one of those, I used to love those TV documentary programs where they used to do like surgeries and, you would literally see into people's bodies. I was like obsessed with those kinds of things. And then when I went to the local comprehensive school, and when I first started the comprehensive school, my dad got diagnosed with throat cancer. So it was a pretty tough year, but actually what was, I say, quite cool about it was that my dad ended up having this really sort of invasive operation, which led to him having what is known as a laryngectomy. So he basically has a hole in his throat that he breathes through. Now, to anyone, this sounds a bit odd, but this was fascinating because you could take a torch and literally look into his body. And the research nurses that used to look after him were like amazing. Like, he would go for all sorts of things and they would get me involved in his, in the types of things that they were doing and sort of explain it to me. And I think that sort of really helped that whole process. But that really sort of, I guess, hit home for me about the really sort of like fascinating and amazing ways that researchers have gone about identifying ways to help patients with cancer. Not necessarily just drugs, but just, the various ways that we can treat patients. So I went through school and I loved the sciences. I was curious, constantly asking why. Knew that after everything that had happened with my dad, that I was interested in being close to the clinic. So what we call something like, that translational side of science. But at the same time, there was something about research. I didn't want to be, just a doctor sort of treating patients with, the drugs that were already available. I wanted to be part of that process of finding new ways to treat patients, better or in more appropriate ways. So I knew that I wanted to do research. Being from a very small village in Wales, I spent many times looking through prospectuses of universities trying to decide what was the most appropriate degree. And I found that actually really hard. There's biochemistry, there's pharmacology, there's molecular biology, there's genomics, there's just straight up biology. And actually, if I had my time again, I'm not sure if I would have done pharmacology. But from reading those prospectuses, pharmacology ticked a couple of boxes. It was very much about testing drugs on organ systems. There was a lot of stuff about toxicology, side effects. It all seemed to tick a few boxes. So I ended up going to the University of Portsmouth to do pharmacology. I had a great time there. It's right by the sea, made a wonderful group of friends. And as soon as I got to Portsmouth, one of the things that I decided upon was that I really wanted to take a year out of university and spend a year in industry. This was really important to me to really get a feel for like what research meant, not just from doing lab experiments at university where you sort of get told what the answer is supposed to be and you just have to go through that process. I wanted to be in a very real research setting. So I was really fortunate to get a 12-month industrial placement. And the one key thing I'll point out here is a lot of people go to university and think they can coast their first year. You can't coast your first year if you want to do a 12-month industrial placement, because your first year marks mean something. So, so that was one key difference between me and a lot of other people that were reading pharmacology at the time, was that I really cared about my first year marks. So I ended up going to Sandwich in Kent, so still in the UK. And at the best year, it was like phenomenal. There was 50 of us at Pfizer across all of these different therapeutic areas, working on a range of things. I had like the most wonderful PI, Frank. He'd never had an industrial placement student before. He had no clue what he was doing. And we sort of trundled through it together, and it was just amazing. And I actually met my now husband at Pfizer. And then, so went back to Portsmouth, finished university. Pfizer had sort of really, I guess, hit the nail on the head for me that research was something that I really loved, and I knew I wanted to do a PhD. And then by accident, really, I ended up going back to Wales, so back to Cardiff University to do a PhD, and it was part funded by AstraZeneca. I think one of the things that really hit home for me when I was at Pfizer was really being able to do academic research, but with that translatable aspect of having pharma companies involved that are generating the new drugs that are going to go into the clinic. So that was really important. So I went to Cardiff University, and I was basically reading, I was studying a project about looking at resistance mechanisms to breast cancer drugs. And I really enjoyed it. And my husband, well, my now husband, had decided that he was leaving science. He became a chartered accountant. The plan was, as Patricia said, science gives you this wonderful opportunity to go abroad. And the plan was that I was going to go and get this postdoctoral position in Boston, and he was going to move within the company. And then, you know, classic life, he decides he hates accountancy and he's applying for a PhD at Cambridge. So we had the usual thing. So I guess I just wanted to say, you know, life isn't perfect. We can make our 5-year plans, but there's always sort of something that can sort of, you know, drop in the way and you have to make a decision. And, you know, Cambridge, as Patricia sort of pointed out, is not a bad place to do science. So, you know, it wasn't like you picked somewhere awfully terrible with no good science going on. So I decided that I would investigate both and make a decision. And I ended up at the Sanger Institute. And it's been by far probably the best decision of my career coming to the Sanger Institute. And so when I first came to the Sanger Institute, my role was to take a new technology, which is basically a technology that allows you to grow patient cancer samples in the lab in order to test drugs and to do different types of experiments. So with the ultimate goal of trying to identify new therapies for the clinic. And we had some really good success in the first 3 years of that project, which led to us sort of expanding the project and getting funding from Cancer Research UK. And that allowed me to move from a postdoc position, which is basically, you know, you work by yourself and you have a very defined project and very defined deliverables, into a staff scientist role, which was more about building a team of people who could deliver on this bigger project. So it was much more about strategy and long-term impact of your project and how are you going to make this happen, you know, rather than just coming in and doing the doing bits. So this is an example of what I do. So this is a piece of tissue from a patient that had pancreatic cancer, and they kindly donated some to our study. And basically using the technologies that we have in the lab, we're able to basically take the cells and grow them indefinitely in the lab, and basically test drugs, and people like Patricia can take all of that data that we generate and see if there's anything meaningful in the drugs that we test. So I've been doing this now for quite a few years. and I absolutely love it. It means that I get to interact with hundreds of people. We've built like these huge consortiums across the UK and across the world to acquire patient samples in order to generate the best models. And by the best models, I mean, for patients that, really have cancers that are not very well treated. So we can make those models in the lab and try and identify the most appropriate treatments for those patients. And then I guess I have a slide very similar to Patricia's about why I love my job. So the people, this is quite an old photo of the lab, and these are just the people that I work with within Sana, and it doesn't even represent the hundreds of external people that we get to interact with, So as I said, all of our collaborators, we do a lot of stuff with the public engagement team to interact with even patients, for example. We're constantly learning. No day is the same. And that's probably one of the things I most love about my job. There's always a fire to fight. And it's just wonderful. I came to the Sana Institute not really knowing much about genomics or sequencing. And now it's my bread and butter, as we say in the UK, which I find quite phenomenal. There's always this potential that the work that you're doing will ultimately allow you to help a patient. And, we do quite often forget about that when you're doing your day-to-day job, but it's always very important to come back to that point that, this is why you're doing what you're doing. There's a variety of skills. You're not only a scientist, especially if you go into this role of a staff scientist role. We project manage, we lead, we problem solve. There are a multitude of different jobs that we could go into if we decided to leave science and be more than well qualified for. And lastly, I have a really great boss who's allowed me the space to sort of explore this project and make it what it is. And the Sana Institute has been a really wonderful place to work. So I'd be happy to answer any questions that anyone might have.
Awesome, thanks so much. I'm just going to see if there's any questions. Do pop them in the chat box for the Q&A. To start with, I'll wait to see if any come in. You said that you did a degree in pharmacology, but actually if you sort of had your time again, that might not have been the one you'd have chosen. So what advice would you have when, and I think a lot of us felt the same, I felt the same, that there's so many degree titles that are all very similar. What would be your advice if someone's interested in biology or sort of biomedicine, how to choose a degree or pick a degree title when they're also similar?
No, I think that's a really great question, Em, and I don't think I have the perfect answer. I think, one of the things that is clear to me, even things like what we're doing right now, just exposes people to the potential opportunities that are out there, and I would be more than happy if someone was interested in, I want your job, what I think would have been the most appropriate undergraduate degree to have done. I think we're at the same time, I think when you go into a STEM subject or even a biology subject, there's always room to navigate away from that. There's these core principles and core skills that get instilled in you in your undergrad and throughout your career that allow you to move between these various places. But yeah, I think it is hard, when you're finishing, high school and you have to make that big decision about what you do at university. And I think coming along to things like this evening and just talking to people are more than happy to share their experiences is probably the best that you can do.
Awesome. And then there's been two specific questions for you. One linking off that that degree question of, if you could go back again, what degree would you have picked instead of pharmacology?
So pharmacology is an interesting one because you basically have like whole organ systems that you do test drugs on, basically. Whereas actually, I was far more interested in molecular biology, and the DNA and the bits and pieces that make us up and mutations and genomics. I probably would have gone down that aspect of things. But I also think that broader degrees like biochemistry are actually really good for getting fundamental principles across a broad range of subjects that really set you up in a really good way to go on to a career in research.
Awesome. And then one more specific question for you. What do you think would be the next step in your career journey?
Oh, it's a great question.
Big question.
If you know the answer, could you please inform me? I'm not sure, to be honest. I think I mean, to be honest, I think being in academic science is quite tough. There's generally one trajectory that people think about, and that's becoming a group leader, and having your own group and that kind of, and that is not something that I want for a multitude of reasons. But as I said, at the moment, I have a fantastic job that gives me the space and the breadth to do all the things that I want to do. Like Patricia said, I think moving into industry is just like a wonderful place to be now. And then there's also like a multitude of jobs outside of that, about project management, within charity sectors that are trying to set up really big scientific projects and require someone with leadership and project management skills, but to really understand the science as well. So we're perfectly poised to go into those kinds of roles as well.
Awesome. That was all the specific questions for you. So thank you very much. And we'll take any extra questions for Hayley at the end. But next up is Jose. So do you want to share your screen and share your story?
Okay, can everyone see?
Yeah, that's great.
What I'm sharing? Okay, perfect. My name is Jose. I'm from Portugal. It's where I was born and raised and spent most of my days. And basically, yeah, I'm a PhD student at the moment, and the PhD is a degree you can get, which basically means you're a doctor of philosophy. And what this means in today's terms is you're a producer of knowledge. And what this means in simpler words is that you just did research and you found out something new and you wrote a thesis by the end of it, which kind of explains what you did. And this is what I'm doing now. I've been doing bioinformatics, more specifically within the realms of cancer data science, and I'll be getting into this. But first, I'll just try to cover a little bit what I did. So I come from Portugal and I did the science bits of high school, basically. So everything very normal. And then I went, in my last year of high school, we kind of have to pick a few subjects, and I picked biology and chemistry. And then intuitively, I went to study biochemistry, which feels like a very logical step from those two, at least. I went to the University of Coimbra, which is similarly to Cambridge, very, very old, and people enjoy dressing in weirdly old attire when they go to the university. And this is what I'm wearing here. The hat was not included. It was my own addition to this basically sartorial choice of me. Basically in biochemistry, as interestingly as was mentioned before, you kind of get a lot of really good basis for a lot of basically a lot of what is in the end molecular biology. So my biochemistry degree was a bit more traditional, so it kind of involved quite a bit more chemistry. And during this, like my education, I was also involved in a number of other activities, such as I was involved in the student union and organizing a few activities there. I also did quite a bit of radio. So in Coimbra, there's one of the few amateur radio schools in the country. And so I kind of had the opportunity to join it as well. And also during this time, I also with some friends, when I started to getting more into the more data science-y world, we kind of founded, or I co-founded a small junior enterprise, which is kind of like a small company, which gives students an opportunity to kind of get more involved with how it is like to work at a company. So it's kind of like a part-time job and you don't receive anything for it, but it's very good fun either way. And you get some really nice experience for the future. Now so this kind of doesn't really explain how I went into bioinformatics. And in my bachelor's, I started doing kind of a few courses in programming. A few of them were offered by my university. And I was like, okay, this looks fun. It's not something that I can imagine myself using, or I couldn't imagine myself using at the time, but sure, let's go for that. And then in my master's, we had these lab rotations as a part of the degree. So we could go for like 2 months for a specific lab. And this is when in my first one, I actually figured out, well, this isn't really for me at all. I don't really enjoy being in a lab. It's too finicky. I'm too messy as a person. And I get very nervous thinking, oh, did I add this reagent already? Did I do this? Have I done this already or not? And so this really wasn't for me. And so I started looking into this new thing, you know, bioinformatics, or to me, it was a new thing. At the time, it wasn't a new thing at all. And so I kind of took what I had from my programming courses in my bachelor's, and I started to look into this. And then I did a position in a mathematical modeling laboratory. And then finally, I landed in the laboratory where I did my master's thesis, which was in so bioinformatics intuitively, more specifically in just protein structure and associated with prediction and kind of trying to figure out how proteins interact with each other, but from a computational perspective. So using massive amounts of data, which are already available, and trying to do something with them. Then immediately after this, after finishing my master's, I got a PhD position at the European Bioinformatics Institute and at the University of Cambridge, which I'm now doing at the moment in this field, which is cancer data science, which I can probably specify it a little bit better by doing a very simple deconstruction exercise. So cancer data science, you have cancer, a disease which is best described as an uncontrolled division of cells in your body, which shouldn't be happening at all. You have data, which is just a collection of facts, statistics, numbers, quantities that are all kind of unstructured or structured. And then you have science, which is a systematic and practical activity that tries to basically produce knowledge through experimentation, generally about the natural world. And this is kind of what cancer data science is. You have a lot of information about cancer, which is either available or is made available for you by experimental biologists or experimental biomedical researchers. And you can use this in a myriad of ways to actually better understand how cancer works, what causes cancer, and what is caused by cancer in terms of symptomatology, which can also aid clinicians in the day-to-day. Now, specifically and regarding what I actually do, so I have two main projects which I've developed during my PhD. The first of which is here, very simply described as looking at blood. So in the day-to-day, a hematologist will have to kind of analyze their patient's blood if there's something wrong with it. And in cancer specifically, diagnosis can be a bit variable between hematologists, and it can be quite complicated to detect specific shapes of cells that are floating around that can be indicative of specific mutations, for instance. And this is kind of what I spent a lot of my PhD doing. It was kind of automating this process of analyzing these little cells, trying to identify what's going on with them, and if there is any relevant association to specific mutations or to specific cancers or specific cancer subtypes. What I've been doing more lately is trying to understand mutation evolution. And what I mean by this is, so we can sequence people. So it's not quite a popular thing. You can have yourself sequenced as well. But what we did here was to sequence quite a few very old people, so very senior individuals. And by doing this, what we get in the end is, along several years, we have like this group of sequenced individuals, and we can see how mutations evolve in their blood. And this is what I've been doing as well, trying to see, okay, there's a mutation in this gene specifically. How does this mutation evolve? Is it because it's in this gene? Is it because this person specifically has something going on with them? We can try to see if their blood levels or some blood parameter they have, like a white blood cell count, is altered, and try to find some nice correlations and associations there. And now this is something that I forgot to mention, which is the reason why we do this. It's not necessarily related to cancer, this bit here, so understanding mutation evolution, because we're just looking at healthy individuals. Because the scary truth of being a person is that you're constantly accumulating mutations on the background of your life. And most of these are kind of harmless, but they still grow. And this is kind of what we're trying to do here. And this is important because some of them, which are the ones which aren't really harmless, they may develop into a cancerous development, which is the nastier bits of it all. And this is pretty much what I've been doing through the last years of my life. And just to leave you with something that you can see, which are quite interesting little factoids about cancer. You kind of have this first one, which is the immortal cells of Henrietta Lacks, which tells the story of how probably the most popular cancer cells came about. So the HeLa cells, which might be familiar to some of you or not, and how they actually came about. And also this little video about Peto's paradox, which is looking at how very large animals apparently don't get cancer, which is just something that's been interesting scientists and cancer scientists especially for quite some time now. And some people are looking into it. And all of this is really quite interesting. And I guess I just forgot to say why I'm doing a PhD, which is I just really like to learn. And it's a unique opportunity for you to actually continue learning. And it's quite a unique environment as well. It's very stimulating. You're just given a problem that you have to solve. And by solving this problem, it's not just the gratification of being able to solve a complicated problem. It's also the gratification that you solving this problem is actually helpful, or has the potential to be quite helpful to improve a lot of people's lives. This is pretty much it. I don't know if anyone has any questions.
Awesome, thank you so much. Yeah, if anyone's got any questions for Jose or for any of our speakers, pop them in the chat box and we'll pull out some to ask to sort of all of them together. But so you're doing your PhD at the moment. What might be your next sort of steps? What options might you have after your PhD that you might be thinking about, or maybe that you don't know yet, but what options would there be after a PhD?
I would be really glad to tell you this if I knew, but I really don't quite know at the moment. So it'll probably involve still working with, just working in a data science related field, probably in computational biology. I'm also quite interested in data visualization, which is something that kind of grew on me over and during my PhD. So that's also something I'm trying to explore a little bit more now in terms of employment possibilities. But yeah, I think this is it.
Awesome. And we often have a lot of people who maybe are interested in coding or don't know like where to get started on sort of coding and data science. So would you have any recommendations of if someone doesn't know anything about coding, where to sort of start?
Where to start? I mean, I'm not going to recommend a specific tutorial or anything. I think there's so many of them that it's mostly religious, which one is the best one or the worst one or whatever it is. But if you really enjoy it, if you just Google, so Python would be the usual introductory language for everyone. If you Google Python tutorials and try to go for something that actually also makes you solve a problem. Like so I have programming classes in uni, and that was pretty cool, but you don't really have a problem that you could solve in your mind. So the first time this actually happened was when I was still in an actual laboratory, and I was like, okay, this repetitive task is really, really boring. Is there any way that I can stop doing this very repetitive task and kind of automate everything? And this was my actual first experience. So it's really quite more easy to just learn programming if you just have like a problem in mind that you want to solve. And I think that's kind of something that is usually missing when people start learning how to program.
Awesome. We'll bring Patricia and Hayley back onto the screen if I can find the right buttons. There we go. Spotlight. There we go. And so if anyone's got any questions more broadly about anything that any of our speakers talked about, or just more broadly about sort of going into cancer research or sort of biology in general. Someone asked a question earlier that I think was for Patricia, but actually I think would be sort of suitable for anyone, asking whether your job includes chemistry as well as biology, and sort of what proportion is chemistry or biology?
I would say, well, it definitely contains both. It depends a little bit on the project that you're on. For me, looking obviously into drugs and drug responses in cancer cells, I have to understand the biology behind it. So why would a certain drug work in a certain cancer cell, but not in another, for example? Chemistry can't hurt, but I have to say my chemistry knowledge is rather basic. So it's not as if I would know the structure of the drugs, for example, but it would certainly be helpful, and particularly moving into industry, I think in drug research, chemistry would be very, very valuable. So you can come from either direction. I think just having that understanding of how you process things, how you learn things, you can learn other things on the way. So if I don't know something, I'll Google it, or I'll find a book that explains it to me and I'll just read it.
Awesome. Hayley, does your job involve much chemistry?
No, it doesn't. My husband is a chemist by training. So he did chemistry at Bath. And if I sat here and said that my job included a bit of chemistry, I can only imagine what he would say to me. But no, so same as Patricia, I did chemistry at A level, and then going through my undergrad, there was some chemistry, but the major focus was biology, and that's really what it continues to be.
Awesome. Someone on a similar sort of topic has asked whether physics has been involved in any of your careers.
I'm glad it isn't. I have to be honest. It was not the worst topic to study. So I had it in my bachelor's. It was part because I did molecular life sciences. I had everything: chemistry, physics, math, biology. Biology was the one that really rang a bell for me, and that came the easiest. That's why I went into that direction, I guess. But at the same time, many things use physical principles. So if you know them and if you understand them, that is always really, really helpful. I think it's just a slightly different way of thinking if you come from that direction, but it is a very valuable one if you enjoy it.
Yeah.
Awesome. There's also the sort of whole field of biophysics, which obviously very much uses physics to understand biology. So I think one of the nice things about biology sciences is that you can use other bits of science to sort of look at biology. On a similar note, we've got questions in the chat box and in the Q&A. Someone's asked, and it might not be something that any of you know, given the routes that you've come into science, but they've asked whether you can enter research after completing like a medical degree to be a doctor. So I don't know if any of you have experience or know colleagues that have done that.
Yeah, most definitely. Especially within the cancer program at the Sanger Institute, there's multiple PhD students who are clinicians, who have decided at some point along their training that they would like to sort of, take time out to investigate a specific question and come and do that. And yeah, I think it's phenomenal actually, because they really understand what they're treating, if you know what I mean, and they're able to bring that knowledge back into a research group where many of us have not interacted with the patient, even though that's what we're trying to do. We're trying to help those patients. So yeah, certainly, and it's actively encouraged, and there are lots of programs out there, funded by CR UK, as well as other charities that really encourage clinicians to take that time out to come back into the lab and do a PhD.
And it's very common in German-speaking countries as well. And if a clinician decides to join a group, I mean, everyone wants to have them because of their very specialist knowledge. And it's very good to see that they can share their knowledge as well with other people. So yes, definitely.
I'm on mute there. There's been two more specific questions about picking sort of areas to study at a slightly higher level. So we'll start with the one about masters. So someone said, in order to get into drug design or drug design for cancer, which of the following would be more useful at a master's, looking at just cancer biology or biochemistry? Patricia, I imagine you're probably the best place to sort of answer.
I would say cancer biology is often part of more basic bachelors as well. So you could gather quite a lot of knowledge in your bachelor's already. Whereas if you're specifically interested in drug design and so on, I would really specialize in that in your master's. So that is how I see the master's. You can really hone in on something that you're interested in and go for that. I unfortunately didn't. So molecular medicine was not specialized enough. I had a great time, but I knew I wanted to do cancer biology, and I wasn't brave enough to go for cancer biology. So I think if you know what you want to do later on, then find your master's that really matches that if you can.
Awesome. Haley or Jose, either of you want to add anything to that?
I guess in that topic, it is a bit complicated. I mean, for drug design, I mean, what I did in my master's was tangentially related to drug design, and I guess it will really depend on which aspect of the drug design you'll be mostly involved in. If it's in doing cell assays, you'll probably be happier knowing a bit more about cancer biology. If you're a bit more involved on the actual drug design and just screening for possible compounds, you'll probably want to be better trained in biochemistry, or quite a bit better trained. So it really depends on what you see yourself doing in the future. If you have, if you like one stuff more than the other, just go for it at that point.
And then sort of on the other side then, so I think that person sort of quite clearly knew what they wanted to get into. Someone's asked, how would you recommend sort of finding out which specific area you might want to go into, thinking about sort of master's or PhD level? How did you know that that was the specific area of biology you were interested in?
I think it's really wonderful these days that lots of research institutes, including Sanger, now offer with the PhD programs this ability to do rotations, so you can sort of test labs out, and that you want to be involved, if you come to Sanger in some sort of genomics, but you're not wed to cancer, or you're not wed to pathogens, for example. This really does give you an opportunity to try all of those things out and try different teams out, which I think is really exciting, rather than having to make that really big commitment at the start and then sort of be in that lab for 3 to 4 years. So I think it's not something that I did, and thankfully it worked out for me, but certainly speaking to people who've been part of these rotation programs is certainly something that they find really, really useful in ensuring that they make the right decision for their PhD.
I've mentioned it before, but if you can do an internship or speak to someone who does the job that you think might be interesting, go for it. And in German-speaking countries, you don't really have rotations ahead of your PhD. So you have interviews, and you might speak to quite a lot of PIs, so group leaders at the same time. That is how I got my PhD position, which was really good. So I could compare them, and it was really just gut feeling, like this sounds interesting. And then I went for it. And thinking back, I might choose something else now if I had the choice again, but it was still a good choice. And to me, it feels sometimes as if the PhD system in the UK is quite rigid, as in it's a prefixed 3 to 4 years, and you can't really deviate from that. Whereas in other countries, it's a lot more flexible. So if in a German-speaking country, you start your PhD and after a year you decide, oh, this is really not for me, I'm going to go for a different project, you can. So it's a little bit, it's different, it's a different system, but you can always be flexible. I would say, rather than being unhappy for 3 or 4 years, please be brave enough and just go for something else, because it's not going to make you happy, and it's not going to make other people happy around you either.
I think the one thing to add as well is not just about the topic, it's about the lab. And that's equally as important as the topic, and being surrounded by people and a PI that you get on with, and you can be the best that you can be. So that can be a difficult one to judge, and is why, as Patricia was saying, it's really important to talk to as many people as you can from that lab to get a really good feel for how they operate, because every lab is different, even within one research institute.
I think that extends sort of into other areas as well. If you're looking at colleges or sixth forms or universities, it's not just what you're studying. Do you like the place, both in terms of, Patricia, you were saying that the UK is a bit flat for you. Do you like geographically where it is? Do you like the actual spaces that you'd be using? Do you like the people that you're with as well? Because you could have on paper, the best degree on the planet, but if it's not something that you're happy with, it's a long time in your life to be spending doing it. We've had a couple more questions. Jose, one specifically for you, asking whether there's any new information or breakthroughs that you've had from your work in mutation evolution.
I'll try to not be very technical about this, but so we have kind of a unique cohort or a unique group of individuals because we have longitudinal sequencing, and what this means is we have readings for the fraction of mutations in a specific gene each individual has for a few time points along 10 or 20 years. And so what this allows us to do is we can kind of see when these mutations are actually appearing. We can see if this mutation evolution is kind of slowing down with time as well, which is something we're observing. We can also, apart from this, we can confirm this kind of with other techniques, which kind of also help us. And we can kind of, so basically, I guess the main findings would be that we are observing kind of mutations whose evolution kind of slows down later in life. We're still a bit confused about why this happened. It doesn't seem to be driven by anything we can easily explain for now. What we're also seeing is that a few mutations are only acquired after you kind of turn 40, basically. It's kind of the deadline. So the general consensus is that mutational processes happen consistently through life. So you kind of acquire mutations at an equally, at an equal rate through your life. But for a few genes, this doesn't really seem to be the case. So you only have mutations appearing at really later stages of your life, and they grow really, really fast. And what's really fascinating is that these individuals have these apparently very aggressive mutations, but they're just healthy, just perfectly healthy individuals, which is great news for them, but it's very confusing for us studying. I guess this would be the main ones.
And we've had one last question that I think will be a nice one to wrap up on. Someone's asked, what are the most essential skills to do research? So what skills would you say would be the best ones to develop if you're interested in going into research?
I think you have to be resilient, because nothing ever works out the first time you try it in the lab, for example. So if you can cope with that and just blow on and try it again, and you're in the right headspace to do that, that is a very good trait to have.
Yeah, I think you have to be curious. You constantly have to be asking the question why, and not be, always wanting to like push it further. And then I guess, just practically speaking, and I think Patricia mentioned this previously, given the times that we're in right now, and not COVID times, just the generation of lots of data. I think if research was somewhere that you really wanted to be, I would definitely encourage people to get coding. It feels like every job within research will require some aspect of coding, and it's just something that you should definitely jump on if you have the opportunity.
Awesome. And Jose, any essential skills that the others haven't already touched on?
No, I would say, like, in terms of hard skills, like probably statistics is something that you'll use. It will be abundant in your research life. In terms of soft skills, just, kind of having a good mindset that doesn't, just know that when you're doing research, you're not competing with anyone. Is the key thing to know, because otherwise you end up comparing yourself to unreasonably successful people. And that's not good for you. So you kind of just go for what you've decided for yourself, stick with it. Maybe it works out for you, maybe research isn't for you, maybe it is. But if you're constantly comparing yourself with other people, that's kind of the recipe for a bad time in general.
Awesome. I've got some links for some coding recommendations from one of our previous speakers that I'll pop in the chat. And again, I'll stick it in the email that you'll get tomorrow. That brings us up nicely to the end of our session. So thank you all three of you for joining us this evening. Just as we finish up, I'll mention that our next series of Genomics Lite will be on biodiversity and evolution. Again, we'll have 4 of these running through June and July. Speakers and tickets are yet to be announced, but if you head to this website, there's some information on what we'll cover in total, along with some learning resources if you want to start having a look at what we'll be covering in the series. Other than that, thank you very much everyone for joining us this evening, and hopefully we'll see you for our next series in the future.